The seed structure. Now, the first process in the growth and development of most plants is seed germination. So we first start by looking at the structure of a typical seed. A typical seed consists of the following parts. There is the embryo. Remember, the seed develops from a fertilized ovule. And after fertilization, the various parts of the ovule gets transformed to form the seed. The outer covering of the ovule, that is the integument, becomes the seed coat. The triploid nucleus that is formed after the second fertilization and forms the endosperm nucleus becomes the endosperm of the seed, while the zygote becomes the embryo. So the embryo in turn consists of the plumule, that is the embryonic shoot, the radicle, the embryonic root, and one or more cotyledons, which is for food storage. The number of cotyledons depending on whether the plant is monocotyledonous or dicotyledonous. Then there's also the endosperm. In some seeds, the main food storage structure is the endosperm. Such seeds are said to be endospermic. For example, the maize seeds. In other seeds, the endosperm is absent, and such seeds are said to be non-endospermic. The endosperm gets absorbed into the cotyledons during development, and that is why at maturity, the seed does not have an endosperm. For example, the common bean seed. Then there is the seed coat, which, as shown here, develops from the integuments. It is a protective outer covering for the seed. It consists of the outer thicker testa and the inner thinner membranous tegmen. So there's a tegmen, which is the inner membranous layer, and the outer thicker testa. The seed coat provides protection from infection by bacteria and fungi, and also from physical injury like mechanical damage. And in many plants, the seed coat also assists in dispersal. A seed has one scale known as the hilum, which represents the point where it was attached to the funicle in the fruit. The funicle, this is a funicle, so the point where it was attached, this one develops as the hilum. The funicle connected the seed to the placenta on the ovary wall, from which the seed obtained nourishment for development. And there would also be a micropyle. Now we look at the structures of dicotyledonous seed and a monocotyledonous seed to understand the various features. Now, a good example of dicotyledonous seed is a common bean, Phaseolus vulgaris. External features include, you can see the seed coat, that is the outermost covering, the hilum, the point of attachment to the funicle, and then the micropyle, the tiny pore, which is the entrance, which marks the entrance to the ovule. This is the micropyle here. So as the seed develops, the whole remains, and it plays a very important role in germination. Then, for most bean seeds, the radicle will appear as a swelling on the surface. So you can see the position of the radicle. And then internal features, see the two cotyledons, and then the embryo consisting of the plumule and the radicle. The plumule is the embryonic shoot, while the radicle is the embryonic root. Now, there is a stalk that connects the plumule and the radicle to the cotyledons. This stalk is known as the cotyl. Now, between the radicle and the cotyl is the part known as hypocotyl. And between the plumule and the cotyl, there is a part known as the epicotyl that also play an important role in germination. Epicotyl and hypocotyl are growing regions. Then there are two cotyledons as mentioned and a seed coat. The seed coat consists of, there is the outer tester, and then there is an inner membranous tegmen. So that is the structure of a monodicotyledonous seed such as a common bean. Now on the other hand, a monocotyledonous seed such as the maize grain, the maize, looks like this. So the maize grain is actually a fruit. That's why there's still the remains of the style. So externally, you can see there's a small protrusion there that represents the remains of the style. And then there'll be a scar that represents a point of attachment to the fruit wall. Now, internal features are shown here. You have the radicle, the plumule, and there is a cotyl that connects the plumule and the radicle to the cotyledon. There's only one cotyledon. Now, just like in the dicotyledonous seed, between the plumule and the cotyl, there is an epicotyl, and between the radicle and the cotyl, there is a hypocotyl. Now, in addition to there being just one cotyledon, in monocotyledonous seeds such as that of the maize, the plumule is covered. There's a protective covering, a protective sheath known as the coleoptile, while the radicle has a protective sheath known as the coleorhiza. And in the maize grain, the fruit wall, that is a pericarp, is fused with the seed coat. Remember, the maize grain is actually a fruit, but within that fruit, the seed is present and the seed coat is fused with the pericarp such that it is not possible to distinguish the two. So these are monocotyledonous seed with only one cotyledon. Then there is a large endosperm. The main food storage structure in the maize seed is the endosperm. The cotyledon is present, but its main function is more of transfer, conveying of food materials from the endosperm to the growing embryo. Otherwise, the main food substances are stored within the endosperm of the maize grain.